Добрый день, глубоко уважаемые коллеги и гости. Позвольте заседание диссертационного...
Dear colleagues and guests, let me declare open the session of the Dissertation Board of Sergey Nurk, Specialty 0102, Genome Assembly of Non-cultivable Microorganisms. Declare open, and according to the high-performance sequencing data, according to the order of St. Petersburg State University of the 10th of January, the research, myself, Olga Podgorna, Doctor of Biological Science, Professor of Cytology and Histology Department of St. Petersburg State University, has been appointed Chairman of this Dissertation Board. Let me introduce my colleagues, my fellow board members. Matveeva Tatiana, Doctor of Biology, Professor, Department of Genetics, Biotechnology, St. Petersburg State University. Gelfand Mikhail Sergeyevich, Doctor of Biology, Institute of Information Transmission Problems of the Russian Academy of Sciences. In remote mode, we have Skriba Alexander, Doctor of Biology, Bielefeld University, Germany, and Dr. Adam Michael Filippi, Doctor of Biology, the National Institute for Human Genome Research, the National Institutes of Health, United States. According to the federal law of 23rd of August 1996, number 127, on science and the state scientific and technical policy, the St. Petersburg State University has the right to award academic degrees independently. The corresponding order on procedure for awarding the academic degrees at the St. Petersburg State University, on the basis of which we are holding this session of the board, was issued on the 1st of September 2016. Order number 6821/1, hereinafter the order. In accordance with this order, the session of the Dissertation Board is duly constituted if at least 2/3 of the approved members of the board are participating, but not less than 4 persons. Our board consists of 5 members. Everyone is present, including two remote interactive board members. Audio-visual contact with them is ensured, thus there is a quorum. Let me set forth the procedure of today's session with a total duration of no more than 2 hours. The Chairman's brief report on the main content of the documents submitted by the candidate and their compliance with the applicable requirements. Answers to possible questions, 5 minutes. A brief presentation by the degree applicant outlining the key points of his study, 15 minutes. That will be in English. Questions to the applicant strictly on his report, no more than 2 minutes per question, and answers of the applicant. Speeches of all the members of the Dissertation Board, alternately with feedback, with a statement of their positions, questions, and suggestions to the degree applicant, no more than 10 minutes for each speaker. Speech by the Chairman with his feedback. Answers of the applicant to questions and remarks of the members of the Dissertation Board, no more than 15 minutes. The open discussion, speeches of the attendees with a summary of their positions or specific questions and suggestions to the applicant strictly on the topic of the study, not more than 5 minutes per person. To do that, all the speakers are kindly requested to put their names on the registration sheet and introduce themselves in full before speaking. Final answers of the applicant to the questions asked, no more than 5 minutes. Speech of the scientific supervisor. I'll have to read it out, obviously. a 5-minute discussion before the open voting on awarding or not awarding the academic degree to the candidate. For the time of this discussion, the broadcast shall be suspended. Open individual voting. The chairman shall count the votes, and the results shall be recorded in the minutes of the meeting. A decision on awarding or not awarding the academic degree to the applicant. Closing remarks of the applicant, no more than 2 minutes. Before we start the official procedure, let me remind you to switch off your mobile phones. It says here, thank you for your understanding. Let me also inform you that our mission shall be recorded and broadcast online at the St. Petersburg State University website, and the speeches are simultaneously translated from Russian into English or from English into Russian. So, I shall now begin the meeting with the first item. Chairman's summary of the thesis of Sergey for the degree of Candidate of Physics and Mathematics, specialty 030109, Mathematical Biology and Bioinformatics, on the topic Assembly of non-cultivable microbial genomes based on high production sequencing data. Was accepted for defense by the order of the Scientific Secretary of St. Petersburg State University of December the 28th, 2018, number 12923/1. The board was appointed by the order of the Scientific Secretary of St. Petersburg State University on the 10th of January 2019, number 9/1, and I have already introduced its members to you. The applicant submitted the following documents to the Scientific Secretary of St. Petersburg State University. Application to the Rector of the University, Professor Kropotchev, on the acceptance of the thesis for consideration and defense, submitted on the 27th of November 2018. A review by Pavel Arkadievich Pevsner, a candidate of Physics and Mathematics, Senior Consultant of the Laboratory of the Center for Algorithmic Biotechnology at St. Petersburg State University. A list of 3 publications which set out the main scientific results of the thesis, including 3 articles in publications included in international databases and citation systems. A certificate of candidate examination issued on the 9th of November 2017, number 344, issued by the Institute for Information Transmission Problems of the Russian Academy of Sciences, which certifies that Sergey Nurk passed the candidate examinations in specialty 030109, Mathematical Biology and Bioinformatics, History and Philosophy of Science, with an excellent mark, English language an excellent mark, specialism in Mathematical Biology and Bioinformatics, excellent. An Honors Diploma, OSA number 01411, issued on the 7th of July 2011 at St. Petersburg State University, which certifies that Sergey Nurk mastered the basic educational program of high professional education, mathematical support and administration of information systems with a degree, and was awarded a degree according to the decision of the State Dissertation Committee, mathematician programmer. The dissertation in English and Russian languages as a hard copy on the rise of manuscripts and in electronic form. The thesis formatted according to the GOST 7.0 requirements. It should be noted that all the listed documents correspond to item 12, section 3, presentation and defense of thesis, procedure for awarding degrees at St. Petersburg State University, Annex 1 to the order, a brief report of the applicant, Does anyone have any questions? I hate the word "this," and always try to omit it. Does anyone have any questions about the submitted documents? If no, let us give the floor to the applicant for his brief report.
Give you a brief overview of my dissertation project. It is dedicated to developing methods for assembling genomes of non-cultivable microorganisms. Access to the genomic sequence of a microorganism provides biologists with unique opportunities for analysis of its vital processes and is critically important for modern microbiology, ecology, and healthcare. The process of identifying the order of nucleotides in a genome is called DNA sequencing. Rapid development of high-throughput sequencing technologies in the past 15 years drastically reduced its cost, making genomic studies a common scientific practice. Unfortunately, modern sequencing technologies are able to read only relatively short DNA sequences. In particular, Illumina, most widely used sequencing technology, produces reads no longer than 250 nucleotides. Actually, Illumina sequencers typically produce paired reads divided by some distance in the genome. Basic approach to genome reconstruction starts with sequencing large number of overlapping reads and then computationally assembling them into longer genomic fragments called contigs. Unfortunately, conventional methodology requires millions of copies of the target genome. Thus, it cannot be applied to the majority of bacteria and archaea from natural environments, which at present cannot be cultivated or isolated in necessary quantities in the laboratory. Today, genome analysis of non-cultivable microorganisms can be carried out by two methods. First, single-cell sequencing, which uses special techniques, in particular multiple displacement amplification, or MDA, to amplify DNA from individual cells. And second, metagenomic sequencing, direct sequencing of all DNA from a microbial community sample. However, datasets produced by each of those methods pose additional assembly challenges. Study of relevant literature has shown that those challenges had not been adequately addressed before. Thus, the main goals of this work were development and official implementation of the methods for assembling prokaryotic genomes from MDA and metagenomic sequencing data. Following the majority of assembly tools for Illumina data, this work is largely based on the analysis of de Bruijn graphs constructed from the set of reads. De Bruijn graph can be constructed in the following way. First, an integer value k is chosen. Strings of length k are further called k-mers. Vertices of the graph correspond to distinct k-1 mers in the reads. Every directed edge corresponds to a distinct k-mer and connects vertices labeled by its prefix and suffix. Using the Bruijn graphs for genome assembly comes down to viewing this graph as an approximation of the graph constructed over the unknown genome sequence. Note that non-branching paths in this graph, which can be much longer than individual reads, correspond to some genomic fragments. We mostly consider condensed de Bruijn graph, which is the result of replacing each non-branching path by a single edge labeled with the corresponding sequence. Length of the edge is defined as the number of k-mers in the original path. We also define coverage of an edge as the average multiplicity of its k-mers in the reads. Genomic assembler Spades was initially developed for assembling reads sequenced following MDA amplification. While being a major tool of single-cell genomics, MDA brings a number of issues which complicate the assembly. First one is extremely uneven depth of coverage across the genome. The slide shows coverage plots for two E. coli datasets. As you can see, in MDA case, coverage manifests rapid orders of magnitude changes. Also, many regions produce extremely few or no reads at all. As a result, many genomic k-mers are missing from the reads. Gaps formed in the de Bruijn graph lead to fragmented and potentially error-prone assemblies. It also becomes impossible to select a single coverage threshold that would allow to clean the graph from erroneous edges caused by artifacts of the sequencing process. The problem is further amplified by the second challenge posed by MDA sequencing, formation of chimeric junctions between different genome positions. Once formed, they are further amplified alongside with other genomic fragments, ending up being supported by multiple reads, which makes their detection during assembly much more difficult. Next slides give a brief overview of the features of Spades assembler, which contribute to improved assembly of MDA sequencing data. First, to mitigate the problem of gaps in the assembly graph, Spades uses iterative de Bruijn graph construction approach, first proposed in IDBA assembler. Spades also implements a gap closing procedure. It starts with finding positions of paired reads in the graph. Then, pairs of dead ends are joined even over a small overlap if they are linked by at least 2 read pairs. Various simplification procedures were implemented to remove erroneous graph elements. The resulting graph is referred to as an assembly graph. For example, several original methods were developed specifically to clean the graph of the edges originated from MDA chimeric junctions. Multiple amplification and sequencing errors in regions of high coverage can lead to the formation of quite complex subgraphs encoding many alternative paths, which we call complex bulges. We developed a novel framework to process them using edge projections, which preserve continuity and length of paths in the graph. Additional bookkeeping information is stored during the projection to allow the consistent mapping of reads back onto the processed graph. The basic procedure for processing bulges in Spades considers projections of individual edges onto alternative paths connecting the same vertices and having similar lengths to those edges. Iterative application of this procedure efficiently removes most of the complex bulges. However, in some cases, none of the bulge edges have alternative paths. To process such situations, we later developed a generalized procedure in which multiple edges of an acyclic subgraph are simultaneously projected onto one of its subtrees, highlighted here in bold. Okay, sequences of assembly graph edges can be considered as resulting contigs. However, in most cases, this assembly will be excessively fragmented due to genomic repeats. It can be substantially improved by using linkage information on relative positions of edges of the graph within the genome. Expander is a module of Spades that can combine various sources of linkage information, in particular alignments of paired Illumina reads, for resolving repeats in the assembly graph. With addition of hybrid mode, Expander gains support for linkage information provided by long-read sequencing technologies. Evaluating assembly quality is a non-trivial problem. We consider a comprehensive set of metrics implemented in Quast software. Table presents results of benchmarking of different popular assemblers on MDA sequencing dataset for Staph aureus bacteria. Note that specialized assemblers targeting MDA datasets expectedly improve on the conventional assemblers here. Also, Velvet SC falls far behind Spades and IDBA UD. Benchmarking on 10 more MDA datasets can be found in the thesis. In general, Spades produces more contiguous assemblies with slightly elevated number of mismatches. With respect to other metrics, in most cases, Spades is either comparable to IDBA UD or outperforms it. Spades also performs well on the Illumina sequencing of microbial isolates. Thorough benchmarking can be found in several independent reviews. From the beginning, Spades has been designed as an expandable and flexible platform for prototyping new assembly methods. Eventually, an entire family of assembly tools has been developed based on this platform, including MetaSpades assembler for metagenomic sequencing data. Technological advances turned high-throughput metagenomic sequencing into one of the main tools for analysis of the microbial communities. Absence of reliable methods for reconstruction of long genomic fragments remains one of the central problems in computational metagenomics. Here is a brief overview of the metagenomic assembly challenges. First challenge are highly variable relative abundances of different organisms in the community. Second, organism populations in the community can be represented by several closely related strains. While genomes of different strains are highly similar, they may also have substantial differences. Taken together, those differences greatly complicate reconstruction of long genomic fragments. Third, even genomes of different species may contain long regions of high nucleotide identity, forming so-called interspecies repeats. Fourth, significant challenge is in processing many metagenomic datasets is their size and complexity. MetaSpades is developed on top of Spades assembler and addresses various challenges of metagenomic assembly. This slide lists some of the notable changes introduced to the pipeline. Since Spades was designed for assembling MDA datasets with highly uneven coverage, we were able to reuse many of its components with only minor modifications. Considerable efforts were invested to scale Metaspades for analysis of complex metagenomic datasets. While Metaspades is still inferior to the most efficient assemblers in terms of speed and peak memory consumption, its requirements are acceptable for most practical applications. Several modifications were made to reduce the number of structural errors in rare genomes. For example, coverage threshold was fixed on a low value during graph simplification. Instead, Metaspades primarily relies on the analysis of coverage ratios between adjacent edges. Also, additional step was added to the pipeline to validate junctions formed at the intermediate stages of iterative graph construction. Expander repeat resolution module was modified to adjust its behavior to the coverage of the genomic region that is being reconstructed at the moment. In particular, poorly covered graph elements are ignored while reconstructed regions of abundant genomes. While analyzing a mixture of closely related strains, Metaspades focuses on reconstructing longer contigs, even at the cost of some genomic features of individual strains. Many genomic differences between related strains are represented by subgraphs, similar to the ones caused by sequencing errors. It allows us to use earlier developed procedures, in particular the one handling complex bulges, to mask those differences in the graph. Metaspades tries to further prevent the fragmentation of the contigs corresponding to the abundant strains by features of relatively rare strains. Some of the graph edges are thus disconnected from regions of much higher coverage, illustrated by thick lines on this picture. The resulting graph is referred to as a consensus assembly graph. Metaspades also features the strategy to use the removed differences between related strains to improve the assembly of the mixture. Slide shows an example of how the diverged repeat copy in the rare strain links genomic regions across a long repeat. Metaspades implements the following steps. After assembly graph is constructed, we launch Expander to generate a set of strain contigs representing both rare and abundant strains. Then we transform assembly graph into consensus assembly graph, as described earlier. Then we identify paths in the consensus assembly graph corresponding to earlier generated strain contigs. Then, tweaked version of Expander is used to resolve the repeats using both paired read alignments and recovered paths. Our attempt at comprehensive benchmarking of Metaspades against three most popular state-of-the-art metagenomic assemblers across diverse metagenomic datasets can be found in the dissertation and in a original paper. Briefly, testing on well-defined synthetic community of 64 organisms gave promising results. To assess the assembly quality, we used MetaQuast tool. Assembly statistics for 20 most abundant references is summarized on the slide. Overall, Metaspades resulted in considerable improvements in NGA 50 contiguity metric for many reference genomes, while showing similar or lower number of structural errors across wide range of coverage depths. Metaspades also improved on other tools with respect to assembly contiguity on real metagenomic data. Here, it is illustrated by cumulative scaffold length plots for datasets corresponding to human microbiome, marine, and sediment samples. This slide lists the main results of the dissertation, aligned with the earlier stated goals of the project. Main results of the dissertation have been described in 3 papers, all published in international scientific journals. Also, 2 talks have been presented on major computational biology conferences. Both developed tools have been widely adopted within the scientific community, were acknowledged in a number of independent reviews, and are routinely used in the world-leading centers of single-cell genomics and metagenomics research. In the end of my talk, I would like to thank my scientific advisor, Pavel Pevsner, and my former colleagues, both in academic university and St. Petersburg University. Thank you very much, and that's all for my talk.
A little bit early. Is there any questions? Есть ли вопросы?
Does anyone have questions? No, and there are no. I understand. There are no questions. In that case, we shall start reading the reviews of the members of the dissertation board. Can you hear me, dear colleagues? I represent the biological department, and in my review I focus mainly on issues related to biology. Development of new generation sequencing methods given new opportunities for study of various biological objects, in particular in microbiology with high performance sequencing methods. Many issues have been reviewed related to environmental relations and phylogenetic relations. The genome sequencing methods enabled a great number of so-called non-cultivable microorganisms. This enabled us to describe the microorganisms in more detail. And while identification of microorganisms is not a relevant issue anymore, but reconstruction of long DNA fragments connected to certain methodological changes. Today there are two approaches to study a genome of non-cultivable microorganism sequencing, single-cell sequencing, for example, MDA sequencing, and metagenome sequencing, even in the of the whole DNA mix from the microorganism community. The specific feature of data obtained with each method significantly is simplified reconstruction of genome fragments, especially the number of errors is significantly lower for been on, and such method has not been developed very full. So this thesis bridges these gaps. That is why its relevance is obvious. Novelty and practical importance of the thesis. I'd like to say that the thesis by has made a significant contribution to software development for reconstruction of prokaryotic genomes based on MDA and metagenome sequencing. Suggested solutions have been implemented. Genome assembler space and metaspace are being used by the leading laboratories involved in genome and metagenome projects. The scope of work. The thesis consists of 122 pages, contains of introduction, 3 chapters, and conclusion, as well as list of abbreviations, a glossary of terms, a list of reference literature, a list of graphs, contains 12 pictures and 9 tables. The reference literature consists 140 titles. First chapter gives basic information of Polish genome sequencing and genome assembly on the basis of reference genome, introduces the brain graphs and considers the possibility of using them to assemble genomes of dual readings obtained by sequence Illumina, outlines approaches to sequencing and non-analysis results for non-cultivable microorganisms, discusses main challenges of reconstruction of genome segments. Second chapter describes algorithms and technical solutions of space assembler. On the with an emphasis of MDA analysis, sequence compares space with other assemblers used for to assemble bacteria genomes on the basis of MDA sequencing. Third chapter describes metaspace assembler and compares its results to other metagenome assemblers. It should be noticed that such division into chapters is non-conventional for candidate's thesis. Mathematicians recommend that first chapter should be dedicated to critical analysis of reference literature. A second chapter, on the basis of performed analysis, should outline new solutions suggested by the author, and chapter 3 should be dedicated to implementation of the new solutions, a critical evaluation. Biology thesis is also has different structure. On the other hand, the decision of Sergey, Nurk, appears very logical because each section is dedicated to one major issue, and within each issue you may find elements of the traditional thesis structure. On the other hand, this style has its own drawbacks, connected with the fact that it's not obvious which research was performed by the colleagues and which work was performed by the applicant himself. A second issue which I faced, I couldn't figure out. Data says E. coli SC and E. coli MC, page 65. Are these results obtained on the same genotype or no? Which genotype exactly? At the same time, a reference genome contains information in a similar manner, page 68. Sauros. I think it's important to understand what exactly was assembled. Otherwise, looking at the results of comparing different assembles, you can also speak about trends. At the same time, metagenome, the description of data sets is given. If the paper had a section or subsection dedicated to study material, this problem could be avoided. When reading chapter 1, I got the following questions. What the author means by genome term. Does it include chromosome or plasmids as well? Chapter 13 says that in 1977, the first virus genome was sequenced, read in 1995, etc. Why you did not mention plants as somebody plant genetics? I'm interested in this. In the same section here, and Latin and Cyrillic is denoted by small letter K. In the end of the chapter, describe problem analysis of metagenome data. Here should be, I think, one important issue was omitted. The matter is that microorganisms have not horizontally transmitted genes, but homologs, which in one species are found in chromosomes on plasmid, and in third species it's found in both places. Considering the size of certain plasmids, TI or SIM plasmid, and sizes of chromosome, the question of the definition of genome is even more relevant. Chapters 2 and 3 gave me a very positive impression, left me with the expression of one drawback, which I have already mentioned. Otherwise, very good comparison of assembly results with use of spades and meter spades and other assemblers. Though the thesis is quite well written and logical, well, it still has some misprints. The most obvious one, yet these comments do not affect the overall favorable impression of the thesis. In conclusion, the thesis has been done on a high scientific level, meets the world standards in the world of bioinformatics. The work is very important for fundamental and applied science. The results have been justly praised by scientific community. The reason in a recommendation of the thesis leave us no doubt. The main results have been presented at international conferences and in articles in prestigious scientists, including the World Scopus, quoted many times. The relevance of the instrument package, spades, which includes metaspades, is available online. Was the thesis by Sergey Nurk, genome assembly of genome non-cultivable microorganisms, based on data of high-performance sequencing, meets the requirements set forth in the order of 1st of September 2016 on the order of awarding scientific degrees at St. Petersburg State University. And degree applicant Sergey Nurk deserves being awarded the degree of candidate of physics and mathematical science, specialty 030109, mathematics, biology and bioinformatics. Article 11 of the above-mentioned order has not been violated. a member of Dissertation 1, Doctor of Biological Sciences, researcher, professor of Department of Genetics, Biotechnology, Biological Faculty at Peterson University, Tatiana Matveyeva. Thank you very much for your review. Let me briefly answer your comments. First of all, about the non-conventional approach, that about the division. I used to divide my work into chapters. This non-conventional approach was used because I wanted to divide my research into the different aspects of my work. And unfortunately, well, the introduction became so long, I had to turn it into a separate chapter. About my personal contribution, yes, there can be some confusion. And while the key chapters of the thesis contain descriptions of earlier developed methods and procedures which were developed by me and my colleagues. When I used earlier methods, I have always tried to differentiate them from other methods. Speaking of my personal contribution, I tried, when working on my thesis, to describe my methods in more detail. And while the methods where I was not involved, I described them in short. I have also added a section which, in addition to my description of other works and joint research, well, it describes my contribution in more detail. Speaking of biological material that I use, I absolutely agree that if we including a relevant section would be very appropriate. When testing genome assemblers, scientists use data of well-described stems. So these data fully agree with sequencing of the same stem. In case where a bacteria, we use K12 stem. I'm afraid that well, this confusion results when we worked on the Russian text. We omitted a part of a sentence, and the English text actually says it corresponds to E. coli 12, and well, the Russian text has just used E. coli samples. It's also interesting, I think, while we are discussing different stems, I would like to say that sometimes sequencing data, even in well-described isolants, because of evolution of such bacteria in the laboratory, they become more and more different from our assumed reference, and we witnessed this many times. But in the context of this thesis, I would say that these samples, they correspond, the genome references very well. Speaking of the term genome, what does it include? In my thesis, I maybe have not devoted much attention to this issue, but in the context of full genome sequencing and assembly, plasmids should be included and should be considered a part of the genome. And when describing the results, assembly results, which I presented in my paper, plasmid sequence was included in our consideration in QUAST program, but at the same time, plasmid assembly is a separate issue altogether, and that is why coverage will be much higher coverage of the genome and what these issues we have not considered. I would like to say only I would like to say that my colleagues found a special instrument for this, which has been specially created for plasmide assembly. That's it. I'm sorry about using small instead of capital letters. Speaking of horizontally transferred genomes, well, there are homologs. I totally agreed, I agree, and actually I have mentioned them indirectly, because speaking of intergenome repetitions, I'll say that I also mentioned conservative genes, yet I think I should have said we're talking about ultra-conservative genes and not homologs. These we were actually planning to include them as well in our study. The matter is that some frequent level, normal level and conservativeness is necessary here, because different bacteria, for example, 90% similar, in the Brew graph and in context of assembly, these homologs will not form repetitions, because resolution of the graph is higher than their nucleotic similarities. And they will not get in the way of the process, the assembly process. What else have I omitted? Thank you very much. Well, the next speaker will be Dr. Skriba. Can you hear us?
Hello.
Alex?
Alex,
Yes.
do you hear me?
Hi.
And you see, Sergey already told that, but in Russian traditions, the questions should be just in your remarks. Okay, right now, if you have any questions.
Okay.
Thank you.
Okay.
So, yeah, could you please summarize your review, and then I will be glad to answer any questions.
Yes. Okay. So in terms of the thesis, it's about high-throughput sequencing that has become a standard of technology and life sciences today. And although tens of thousands of microbial genomes are sequenced every year, the majority is actually unknown because it cannot be sequenced in pure culture. So you present these two complementary approaches, which shed light on the microbial dark matter, which is metagenomics and single-cell genomics. You address these highly relevant approaches in your dissertation thesis and present novel assembly approaches to obtain the genomic sequences of these prokaryotic genomes from single-cell and metagenomic sequencing. Both approaches, combined with high-throughput sequencing, have revolutionized microbiology, providing access to genomic information formerly inaccessible. While assembly algorithms have been available for a while, only very few have addressed the specific problems arising from the data obtained by metagenomics and single-cell genomics approaches. While sequencing isolate genomes generates relatively even coverage of the genome, just as you have shown in your presentation, the multiple displacement amplification used for amplification of the single-cell DNA introduces a highly non-uniform read coverage of the genome and also formation of chimeric reads. In metagenomics, the different abundances of the genomes in the sample lead to very different read coverage of the corresponding genomes. You approach these problems by implementing novel approaches for processing sequencing data from metagenomics and single-cell sequencing. So your thesis starts with background work. In the first chapter, you introduce the topic of high-throughput whole genome sequencing and assembly, karyotic genomes. You introduce the main concept of genome sequencing, assembly, assessment of assembly quality, as well as using drawing graphs for genome assembly. Also, the concepts of single-cell sequencing and metagenomic sequences are introduced. In the second chapter, you present SPADES, a novel method for compact representation of the Boeing graphs in memory. And a special focus of the chapter are methods for the construction and simplification of assembly graphs in single-cell sequencing. This includes methods for removing tips and bulges, as well as processing complex bulges in the assembly graph. Also methods for aligning sequences to assembly graph and incorporating information about the paired read linkage are presented. These methods are underlying solutions for the SPADES assembler. In the third chapter, the metagenomic assembler MetaSPADES is presented. Proven solutions from single-cell sequencing assemblies are used to address challenges of metagenomics assembly, including assembly of different abundant genomes and closely related microbial strains. MetaSPADES incorporates SPADES functionality for the iterative construction of the Boeing graphs. Using heuristic modification procedures, consensus assemblies for closely related strains are produced. You benchmark these assemblers, both for SPADES and MetaSPADES, results of the computational experiments demonstrating advantages over competing solutions at the end of chapter 2 and chapter 3, respectively. These benchmarking results against other popular assemblers demonstrate the advancements for single-cell and metagenomic assembly presented in your thesis. So in summary, your dissertation project has demonstrated proficiency in determining worthwhile challenges in sequence analysis and proved the ability to implement algorithms to tackle challenges in assembling single-cell genomes and metagenomes. The presented work is highly relevant, novel, and accurately presented. The thesis itself is well structured and written. The main results of the work has been published in 3 papers, cited as number 7, 8, 9 in the thesis. You proved that the novel approaches you implemented to assembler SPADES and MetaSPADES have been significant advancements in the field of metagenomics and single-cell genomics. The assemblers are highly accepted and widely used in the bioinformatics community. Thus, the dissertation of Sergey Nurk meets the requirements necessary for granting the degree of candidate of physico-medical sciences, specialty 030109, and I support this action without hesitation. The question I have, the 2 questions I have are actually, when you generate this consensus, the consensus assembly graph, is there a way to reconstruct the underlying strains? That's the first question. And the second question is, as you build special procedures for dealing with different coverages, like the different abundances in the metagenomes, how do you deal with sequencing biases? Because the technologies, they usually also have problems sequencing certain patterns, and that will result in different coverage, even in the same species.
Mm-hmm. Thank you for the questions. Answer to the first question is unfortunately no. So when we are doing some transformations of the graph, they are permanent. So we are storing some additional information, but it can be used mostly for further alignments onto the processed graph. And actually reconstruction of longer fragments corresponding to individual strains within the metagenome, it's super interesting topic, which is quite popular now. And I'm also involved in several projects aiming to trying to improve it based on analysis of higher resolution graphs prior to this crude simplification procedures. Looking at higher resolution graphs when we have multiple data sets forming metagenomic series, which might allow us to have additional signal to reconstruct those strains. Or another project is to try using long-read sequencing data together with short-read sequencing data to try to assemble those like haplotypes corresponding to different strains. But so it seemed for us that it is impossible to do based on just short-read sequencing data from a single sample. So we decided like not to do it and do the modifications that, well, in some cases even prevent other people from doing it based on our output. But it's it's more or less, it seems like a lost cause just from having short read sequences from a single sample. So regarding your second question about the sequencing biases, we don't deal with them explicitly, but implicitly they are dealt pretty well because while designing spades, we've never thought the coverage to be uniform because we were aiming at reconstruction of assembling MDA sequencing data. So we deal with coverage gaps pretty well, and also some of the procedures aim to simplify the graph in the regions of higher coverage. And since we were, aiming at like coverage of 1,000-fold, it's totally okay to have coverage like elevated by 2-fold. And I'm not sure that we see higher sequencing biases in like isolate sequencing. Actually, adding to this question, there is also a very important moment which we haven't studied in depth yet, which concerns differences in coverage in metagenomic sequencing data due to the organisms being actively replicated, and then you have increase of coverage the closer you come to the origin of replication. It's a super important question, but again, it gives more problems not for the assembly but for the further binning of the obtained contigs. And it is not dealt in any way within the community, so it's an open problem for anyone who would like to account for this bias. Okay? Thank you.
Okay, thanks.
Are you satisfied with the answers?
Are you satisfied with the answers? Please say yes if you are.
Yes, yes.
Okay. Теперь мы приглашаем профессора Липи. Adam, are you with us? Are you ready to represent your review? You'll be the next speaker. Please.
Yes, I'm ready. Thank you.
Да, я готов. Спасибо.
I will give a brief summary on my dissertation review for Sergey Nurk. Hello, I'm a computer scientist who also develops genome assembly algorithms, so I'm very familiar with the computational work presented in this thesis. And I wanted to note that it is extremely difficult to build a high-quality genome assembler such as spades, and Sergey has played a leading role in this project. This is particularly commendable because Sergey has taken on two of the most challenging problems in genome assembly, and that is the single-cell MDA assembly and metagenomic assembly. The spades and metaspades assemblers have collected thousands of citations and have been responsible for the assembly of even more genomes and metagenomes, and this has had a tremendous impact on the community, and the entire spades team is recognized worldwide for this contribution. The thesis itself is very well written and includes a comprehensive introduction of sequencing and assembly. This introduction chapter, I wanted to specifically highlight the quality of its scholarship. It is clear that Sergey has closely studied the prior literature and possesses a deep understanding of it. And I think that has led to a lot of the success of this thesis. The validity of the thesis is obvious. It includes comprehensive benchmarking against other state-of-the-art tools and shows Spade's competitive performance. But perhaps more convincing are the results from independent evaluations from other individuals and scientists that have shown spades and metaspades to be top-performing tools. And this is evident by their rapid and widespread adoption in the field. So in conclusion, this thesis is of the highest quality that I tend to see from PhD students. It meets the standards of the degree, and I would like to congratulate Sergey on these great accomplishments, and I look forward to his future work and contributions to the field. I have two questions for Sergey. The first question is regarding the SPADES infrastructure. It has enabled many other successful projects within this SPADES family, and so I wanted to know what has been key to this success. You know, how is the SPADES infrastructure designed in such a way that it has allowed other students and other postdocs to work so well in this environment?
Okay, so I will try to answer that. The true answer is I probably don't know, because
Я хотел бы сказать
at some places, but I think that the key was nice abstraction of the assembly graph structure, and so nicely engineered primitives of changing it, and also we allowed to add like almost any additional information to the edges and vertices of the graph, which would then be appropriately transformed during those well-defined set of like graph transformations, which allowed us to like incorporate a lot of different additional information, and yeah, I think that's it.
So my second question is similar to Alex's question about the strain diversity. So in your bulge removal algorithms, what typically are these sequences that are being removed? And you know, what's the largest variant that bulge removal might remove from the assembly?
Right. That is a very important and interesting question. So in metagenomic case, those procedures are much more aggressive than in genomic. In case of genomic assembly, there are multiple parameters, and actually there are like several rounds of those procedures with different parameters. I think that so the largest bulge that it would proceed would be around 1 kilobase long, with like difference in lengths of the alternative paths of up to, I think, 100 base pair or maybe more. So it is pretty aggressive, and it can throw out some of the actually even important variants within the homological genes. But if we don't do that, we just do not assemble anything. So we thought that and it seems true that it's more important for biologists to get one of the like variants of these genes within their assembly rather than not getting any of them. But for something like phylogenetic studies of the resulting like metagenome assembled genomes, I would say that like sequences resulting from those procedures are might not be the best choice. But from the point of like functional analysis of the resulting proteins, we are usually okay.
Have you ever thought about, and perhaps you do, is there a way you can annotate the resulting contigs that are output by the assembler with how much bulge removal or how much variation was present at that particular location in the genome?
Right, so we are not doing that, and I would say that it is an excellent suggestion. And it would be very good to provide, along with actually some other statistics and some other information of what was really done in the graph and how certain the assembler is. Then what was assembled is actually, well, either correct or whether it represents a consensus sequence, right, of multiple different strains. So it is not currently done in any of the tools that I know, and biologists tend to like to work with the most primitive output from the assembly algorithms. So, but it would be an excellent suggestion, and I think that something needs to be done in this respect. So maybe the contigs should be somehow prioritized and tiered into different groups based on how accurate they are or how many variants they represent. And maybe actually even we can annotate the contigs with some of these removed variants. At least we could, for example, provide for every contig, we could provide a subgraph that led to the formation of this contig with some of the additional variants preserved. So yeah, I would say that it is a super important question, and the field, not only our work, but the field overall has not spent sufficiently and nearly enough time to addressing that.
Very good. I have no further questions.
Okay.
Thank you.
That's it. You're satisfied. Okay.
Yes, I'm satisfied.
Okay. Let's go further. Михаил Сергеевич.
По-английски или по-русски?
По-русски.
Хорошо.
Please. По-русски.
I've been instructed to speak in Russian. Значит, я проявлю милосердие и не буду рассказывать, в какой главе про что написано.
I will have mercy, and I will not tell you which chapter says what, including the applicant. The Metaspades work was quoted 3,000 times, was published in 2017, quoted about 100 times. Both are widely used. And the thesis does not mention papers co-authored by the applicant, which are used independently. Well, it describes tests. The thesis describes the tests in detail. So this is the end of my review because everything is clear. Speaking of the structure is non-conventional. The introduction is very simple, and in that case, the thesis does not give an overview of the entire pool of assemblers, with which you do not necessarily, don't have to agree. Yet the thesis is very readable and is very user-friendly. You can. Let me disagree with the first reviewer. For me, it was very easy to tell results obtained by the applicant from results obtained by the other researchers. I didn't have this problem. In general, this thesis illustrates the life, which starts with an elegant mathematical structure, the Brownian structure, and then ugly biology comes on stage, and this perfect beauty has to be modified 1,000 times to obtain various artifacts, and the artifacts created and while the thesis is dedicated largely to creation, not of the algorithm in itself, but to creation of a practical instrument. And the fact that the instruments are so widely used proves that the applicant was very successful in resolving this task. I only have one question, which I did not include in my review. How is the procedure of gluing the graph together? How does this procedure work with inversions? Obviously, it should process iterations well. What happens with strains with many inversions. That would be interesting to know. And in addition to that, well, I would disagree that replication, you said that answering one question, replication will make a major contribution or will make things more complicated. First of all, because bacteria do not replicate so often in reality, only in laboratories, and in reality they hardly replicate all. There are strains. There are some specific exotic strains, but this is more of an exception. I don't have any further comments. I have just questions. I'm curious to know this. I don't have any further comments. I have some comments related to misprints and poorly coordinated observations. Well, I have them all, included this all in my review. This is not interesting. And you mentioned this appendix section, which sounds like surgeon's thesis rather than programmer's thesis. The only comment I'd like to make, again, this is not criticism, the iteration problem. Iteration is De Bruijn is called De Bruijn, not De Bruijn. Which, because there's this is the name of a painter. So this is the standard situation, because information experts know Dr. Watson and Dr. Watson, which is the same person in English. Prince Charles, when he finally becomes the king, he'll be called Karl, not Charles. So these are in Russian. These are my very minor comments. I think this is a very outstanding paper. And then I was told to read out the end of my review. These comments do not affect the overall relevance and value of the thesis. The thesis by Sergey Nurk, assembly of a non-cultivable microorganism, meets the main requirements set forth order 1st of September 2016, number 6821 on the order of awarding academic degrees. Principal University, Article 11 of this order has not been violated, and degree I have not seen this order, and I assumed it says that the applicant wrote the work by himself, and I believe that degree applicant Sergey Nurk deserves being awarded degree of candidate of physics and mathematical sciences, specialty 03010, mathematics, biology, and bioinformatics. Well, inversions was like an invitation to further discussion. Speaking of inversions, inversions will not be processed because they form another structure, new edges. They form new edges. They don't look like bulges. With this, yeah, we have a certain. This is a certain issue. We could try to detect them on purpose, but I think the accuracy with this method will not be very high. But in case of metagenome, but in case of a genome, when we use colored graphs, the brain graphs, comparing them to reference, we did something similar, and it worked. So the issue with metagenomes is because it's not possible. You cannot use different fragments, different colors, and you get something very complicated. But especially for such major tasks, we had to integrate this method, which separates the edges, poorly covered edges, from well-covered areas of the graph. And as it happens, in the case of 5% of the population. If the inversion is present 5% of the population, you won't have any problems with assembly. So we discussed this earlier with Dr. Filippi, but this issue should be studied separately. Speaking of replication, indeed, assembler has to work with so many pieces for sequencing. I think this issue is less important here, and in the context of my study, I think this issue can be neglected. But I think this cannot influence when we between the most efficient methods use all the coverage information, area coverage information obtained in different samples, and the methods have been tuned so that in case of some bias sequencing, when a certain area is twice more covered, so they can deal with this, okay? But in case of sequences, if the population grows, fragments closer to original repetition, it will be different from other segments, and in that case, so this is there are no specific methods, but everybody hopes that we will have to fine-tune this so much, and binning is an altogether different sphere, and I hope that I have answered your question. Dr. Gelfand. Dr. Gelfand is satisfied. My review will be the last one, and because I have to praise you reading Dr. Pevsner's thesis, I will drop a big part of my review. In chapter 1, you mentioned Lumin technology and assembly of short reads. I think this chapter can be of interest for public, to general public. I think my students will benefit a lot from reading this chapter. So this is how I personally am interested in this paper. There is a number of misprints. Let me mention only one misprint. The Center of Algorithmical UO Technology is like where you write your own name. You make mistake in your own name. All other misprints. And my review is available on the university website. I will not read it out yet. I have questions. For example, as a continuity metric, you used NGA 50. So why did you use this metric? Is it unique or something? In the introduction, you emphasize the importance of k-mere length. Yet you used it when building a graph, your assembly graph, yet you never say, or you say very little, how the length was chosen in practice. Metagenome data may contain eukaryotic genomes. Have you ran into any such cases, and can they be filtered? In metagenome data, the metagenome data may contain a number of cases where it's possible to cultivate those isolates, and by combining this data, can this help to test metagenome assemblers? And one more question which just came to me. You say in your thesis that these are sequences which correspond to straight paths, and in your speech you've showed some pictures which you did not show in your thesis. Okay, let us start with this question. For example, and this is not a very interesting example because all the straight paths, but for example, one such one path, ATA, TAG, has a peak with degree 1. United in such unity from one crossing to the next, and then we come to the next crossing. And now let me answer your questions. Let me start with the last question. You asked about the use of isolates obtained from the same results. Yes, this is a very interesting possibility if they can be used to for assessing quality, assemble quality. My answer is yes, they can. It seems that in the modern world, because our collections of isolates are getting bigger and bigger, that means that maybe we will not have to identify isolates, but find appropriate isolates in data sets for which there is already an assembly available. So if we can find an accurate method that will help us to understand that we already have a very similar strain and use, for example, we have Dr. Filippi with us, and I think methods used by Dr. Filippi can be very instrumental in this sphere, and that is why, yes, I'd like to say yes, isolates obtained from the same samples can be used. Often obtaining isolates is expensive. Yet soon we will have such a collection, such a data set. Can I add something? It's interesting to look not only on isolates but on enriched cultures. Yes, this is very good idea. I have not used such data. Where we have we deal with sequencing metagenome sample, and then results of sequencing with additional modifications. I think that's a great idea, which will improve quality of assembles and make the analysis more accurate. And speaking of chaotic genomes, yes, we can have. We didn't run into any such cases ourselves. Assembly of chaotic genomes is short reads. Usually the quality leaves much to be desired, and that is why maybe I think we are doing no worse than other researchers, but at the same time we're not much better than other researchers. Speaking of filtration, this is also a very interesting question. Frequently it's possible to filter with the help of a reference genome. We can machine learning. So there are some methods available, but often many people would like to filter DNA before sequencing, and so to remove those fragments which will be so hard to assemble. So this has to do with biological preparation. And maybe we shouldn't discuss it in any further today. Speaking of the length of K meres, this is also an interesting question. The answer, how we chose, I don't have a good answer to this question. In our instruments, we have a certain sequence of K meres, and the last value depends on the length of the read, and it's fixed. So how long is it? It's K55, but I would like to make it bigger, to increase it. 55, this is a traditional number for genome assembly, Illumina data. So this enables researchers to get a relatively simple graph. That's if you have a single genome, then the graph is relatively simple, and where you can use further repetition resolution methods. And in other cases, in many cases, it all ends with the graph, which is not true. And we can resolve repetitions and these other K values which we have used. We use relatively small K values which can lead to various issues. The higher is the K value. And well, we can make a mistake. So in order to avoid this, but formal analysis is available. It speaking of selecting 1K once and forever. These methods are not dealing with iteration graph construction, and well, these cannot be applied to genome sequencing, and that is why. Some sort of magic numbers are being used, but again, let me say again, this is a good question. Speaking of NGA50, well, we have used it basically for historical reasons. Everyone has used it, so we decided to use it too. There are many different metrics. NX graphs are very useful for understanding which assembly has better continuity. On the other hand, unfortunately, the community has not invented anything better than bringing these graphs. We'd like to have bringing these graphs to one single number for one X value. So this is the maximum length. And why 50? Why is this? There is no good reason for that. People when they develop methods, not in scientific papers, they look at. They have more information when they look at the. Thank you very much. I forgot to say the magic words. All these comments do not decrease the advantages of the paper, and qualification of the author leaves us with no doubt. The thesis of Sergey Nurk, the assembly of non-cultivable genomes on the basis of high-performance sequencing meets the main requirements set forth in the order of 1st of September 2016, number 6821/1, on the order of awarding the academic degrees at the St. Petersburg State University, and the applicant Sergey Nurk deserves being awarded the degree of candidate of physics and mathematics, specialty 030109, mathematical biology and bioinformatics. The Article 11 of this order has not been violated. Chairman of the board. Well, what else do we have on our agenda? What comes next on our agenda? General comments of the applicant. Would you like to say something? I would like to say thank you to my beloved wife who didn't divorce me. No, that's not what you're supposed to say. Well, you have to say something in relation to the questions and comments. Okay, now let me read the review. Access to biological information of different organisms is crucial for many fields of research in medicine, biology, genome data. Increase our knowledge of various metabolic processes in the world around us, and research of bacteria can affect therapy, synonym of irritated bowel, diabetes, obesity, etc. The modern technology of high-performance sequencing allow to read great number at low cost, short sequences, reconstruction of genome fragments and assembly. Genome assembly is one of the key and covered spheres of informatics. Unfortunately, most microorganisms cannot be sequenced with the traditional methods. Can be used with the use of single-cell sequencing or metagenome sequencing. Both techniques call for specific approaches to assembly. The lack of available instruments impedes research. The thesis of Sergey Nurk is dedicated to modification improvement of genome reading methods on the basis of single-cell sequencing used with the help of MDA method and metagenome sequencing. Sergey has successfully dealt with his task. His algorithm and technical solutions became the basis for popular assemblers, spades and metaspades. At the same time, his algorithm and software solutions are used in many other projects and in many labs. While working on his thesis, Sergey demonstrated the ability to work independently, to conduct research, use reference literature, and interpret the results. Computational methods used in his thesis prove his profound knowledge and understanding of computer science, bioinformatics, information science. While working on his thesis, Sergey Nurk took part in a number of joint projects at the laboratory of the center, and together with leading Russian and foreign scientists. The main results outlined in his thesis have been published in magazines, quoted by the international scientific community databases, and presented at international conferences by information science. Relevance of his research and amount of information and the thesis by Sergey Nurk on assembly or genome assembly with the help of high performance presented for procurement of the degree of candidate of mathematical and biological sciences meets the requirements and should be admitted for consideration, and the author deserves being awarded the degree of candidate of physics and mathematical sciences, specialty 010309, mathematical biology and bioinformatics. scientific advisor, Pavel Pevzner, candidate of Physico-mathematical sciences, St. Elizabeth State University, professor of California University, San Diego, and director of National Center, National Computational Center, USA. Now, we have 5 minutes for discussion. For this period of time, I am asking everyone to leave the room. What?
How to tell it?
Do we approve?
Do you approve the dissertation?
I approve.
Okay, thank
I-
you.
I approve. Also.
Okay, thank you.
Me too.
Yes. And
That's it?
Sergey. Okay. So we all in one voice
In 5 voices.
approve the dissertation. That's okay.
Yes, do you still need me?
Yeah.
Do you hear me?
Hello, Adam. It
Adam,
seems
are
that
you in
Yes.
touch? Oh.
It seems that you will have to vote again for the translation of this.
You have to
video
say
conference.
I approve again because of translation.
I've seen it. He has said
Yes,
he nodded.
I approve again.
Yes?
Okay, yes.
Thank you very
Yes.
much. Wait a minute. Is it okay now? Молодой человек. It's okay?
We have 4 out of 5, it's quorum. Но не поймаете вы его, он все, он отключился и ушел. Но
We have to wait for another board member, but actually we can do without Alex. Please say again if you are voting for or against awarding the academic degree. Professor Gelfand, yes, all the 4 members of the thesis board voted for. Can you please say yes again? Please
Yes, I
say
approve.
yes again. Yes.
Рано!
Congratulations, Sergey.
Thus, dear colleagues and guests, let me say that I'm counting the votes. A majority of members of the board voted for, nobody abstained, and nobody voted against. The decision to award the degree of candidate of physics and mathematics, specialty 030109, mathematical biology and bioinformatics, to Nurk Sergey Yurievich has been passed. Now let's hear the final word of the degree applicant. Thank you, everybody, for participation, for your involvement. I would like to thank my wife, who did not divorce me. And I'd like to thank my friends, my family who supported me, who devoted so much of their time so that I could continue my work, my laboratory where I work. That's it, I think. The meeting is now closed. Thank you, everybody, for your participation. Thank you and goodbye.